I'm Nelson. I'm going into my second year studying aquatic biology, and this summer, with the help of Lindsay Mark, my mentor, I was able to study the behavioral responses of cultured white abalone to a predator, their sea star, one of their predators that they'll find in the wild. But to get us started, I want to introduce you to the abalone. A lot of people don't know what they are. Because they kind of look like rocks, which is a really good defense mechanism. But why do we care about the white abalone specifically? Well, they're reproductively extinct, which basically means it's highly likely that they'd go extinct without human intervention. How did we get here? Well, I'll first start off with this picture. As you can see, there are huge mounds of abalone shells behind these fishermen. In the 1970s, they were highly coveted for their jewelry and their meat as well. They're really tasty. They're considered the tastiest abalone. Why are we still here today? Well, abalone are broadcast spawners. So they don't mate like humans, so they don't like do the nasty. Instead, what they'll do is they'll excrete either sperm or eggs into the water column and let the water do all the magic for them. And you can imagine if they're already in low densities, as they were after the 1970s, they would have a really hard time keeping up their populations or rebounding from having such low populations. Where are we today? Well, I'm working with the White Abalone Captive Breeding Program, which focuses on raising and spawning new abalone. You can see one of the newly settled larvae in that photo, and we're doing exceptionally well, but that still leaves us the problem with introducing them into the wild to try to revitalize wild populations. Cool. Okay. They've done some prior research with using red abalone to try to see how those abalone would do introducing them into the wild after being captively bred. But white abalone, they've never been exposed to their own predators in the laboratory. So this raises a huge question of, do they exhibit defensive responses to natural predators? And if these responses even allow them to escape their predators? And one last question, which is kind of all on our minds as well, is if they could learn to escape more quickly, because with that information maybe we would want to expose them to predators prior to introducing them out into the wild to try to give them at least a better chance in the wild. So the way we did this is with exposing them to various stimuli. At first, we would take a sponge, a non-biological sponge, and touch the abalone and observe it 4 or 5 minutes, and record each of the behaviors that we saw, and then to have something to compare it to, because we didn't just we wanted to see if the abalone's behaviors would change between a sea star and something non-predator. So that's exactly what we did, and that would help us answer the question if they could, or if they had defensive responses to a predator. But then we repeated this 2 more times to see if their escape times changed. And in that way we could see if they could learn to escape more quickly. Before I get to all the data and stuff, I do kind of want to explain some of the behaviors that we saw. And these are also some behaviors that have been seen in other studies on abalone, not white abalone though. But yeah, so these are Some of the behaviors, the first one is epipodial spreading, which you can see in this photo over here. They do this basically to feel what's touching them. They just kind of want to get a gauge on what's surrounding them. They would also gallop, which you can see in this photo. They kind of just ran away after finding out that it is a predator. They would also shift, which basically is them just being agitated. Like they would still move around, but they wouldn't have any direction. They would also twist, which you can actually see in this video. Yeah, and you can see that twisting actually is a pretty effective way to escape in a predator like a sea star, because it allows them to break the suction from the tube feet of the sea star. We also saw some less extreme responses, like resting and clamping, and we kind of differentiated these two because in the data we can see, actually I can just go straight to the data. So these mild reactions, we can actually see that in the control, which is with the sponge, the stimulus from the sponge. Most of the time they were either just resting, just sitting there, or they were clamped down. Basically, they would hunker down on whatever substrate they were on. But in the exposures to the stars, in these later treatments, they were actually moving a lot more and reacting a lot more. So there is a variation in between a predator stimulus and a non-predator stimulus. Yeah, exactly. Cool. So this graph is a little, not complicated, but it's a little weird. It's a little unorthodox. So down here we have the beginning time of the behavior, and each of these dots is a behavior. And then on the y-axis, we have the percent of the trial spent on that action. So up here, we see a lot of epipodial spreading, basically meaning it would start really early and it would last throughout the whole experiment, throughout the whole observation period. And then, so once we introduce a sea star, they would likely spread their epipodia to feel what was touching it. And as time went on, we would see a lot of shifting, which is why you see all of this conglomerate of orange over here. And right soon after shifting, we would also see a lot of twisting, and the twisting is kind of really quick because it's a really quick 1-2 punch type of thing to escape predators, as you saw in the video. And then all throughout the trials, they would either shift or gallop away. Yeah, so that's kind of typical of abalone trying to escape sea stars. Oops, I highlighted them. I forgot about that. So this was kind of cool, kind of the meat of the project, I think, which was finding out that there was a difference in between the exposure, in between the first exposure to the sea star and the consecutive exposures to the sea star. As you can see here, the mean escape time decreased by a pretty significant amount, pretty noticeable one. But what is also really cool about this is that the range that the abalone escape time, the range of the abalone escape time also was condensed as well, meaning that not only are these abalone escaping quicker, but there are more escaping within this time frame, meaning translating that there is less risk of abalone in the wild when exposed to sea stars, when being preyed on by sea stars. Yeah. So, what did we learn? Going back to the 3 questions that I presented earlier, we learned that they do display defensive responses. those defensive responses do allow them for escape, and that they can also learn to escape more quickly after repeated exposures to the sea star. So what's next? Well, obviously, trying to implement this in the wild population restoration would be key, because even having like a fragment of less risk for these abalone could mean a lot more for their populations. More research also would be needed because we only studied sea stars this summer. So there is a pretty wide possibility of different outcomes for different predators. For example, I threw up an octopus on the predator section of that one slide, so that would be interesting to see. And obviously, continued public outreach. Really quick shout out to the reef. We do awesome work with the public to try to keep the ocean what it is today, and more specifically to this project, keeping abalone or revitalizing the abalone population and maintaining that, so something like this would never happen again. Thank you.